All right, so we're going to be looking at from gut to brain, looking at dysbiosis, inflammation, and some of that hormonal interplay. So the gut microbiota brain axis, let's just start with a little bit of definition. And actually, before I jump in, how many of you in the room, just so I can serve you better in my talk, are very familiar with intestinal permeability and treating leaky gut? Show of hands. Okay, and how many of you feel like you're relatively new to this? Okay, all right. So starting with our definition here for the gut microbiome brain axis, it's a signaling that occurs between the intestines, the microbiome within our GI tract, and our central nervous system. So of course it includes the gut microbiome, and the DNA, which Dr. Pero laid out for us, is very extensive compared to our own human genome. We also have the interplay between the neuroendocrine and neuroimmune components of the gut, so interaction with the HPA axis, with our parasympathetic nervous system through the vagus nerve, and about 80% of the fibers in the vagus are afferent fibers, so from the gut to the brain, and only about 20% are efferent from the brain to the gut. So I guess who's controlling who, right? Then in the HPA axis, our control system for our hormones, which everyone is familiar with, but not just cortisol, but also our sex steroids, our androgens, and our thyroid hormones. There are a number of things that are going to interrupt our GI supersystem in its interaction with our modern world. And so the exposome that's going to create this disruption, processed food, low fiber, food additives, all of these things impacting not only the health of the microbiome, but also impacting the survival of some of the specific microbes. Our sedentary lifestyle has been shown to have an impact on the composition of the microbiome. Of course, our chemicals and xenobiotics and environmental pollutants also impacting the health of the microbes. Infections that we acquire, whether they're acute infections that we're aware of or just a dysbiotic imbalance in our microbes, and then, of course, our antibiotics and our medications. A single course of antibiotics permanently alters your gut microbiome diversity. So there are species that you lose with an antibiotic course that will never be regained in your gut microbiome. It's kind of crazy to think about it that way. So dysbiosis at its base is just an imbalance in the microbiome. So we could have a deficiency dysbiosis where we don't have enough, either enough of the bacteria overall or enough of the good bacteria, the beneficial commensals. We could have an overgrowth of commensals where we're just having an imbalance in the utilization of system resources, so it changes which microbes can flourish. Or we can have an overgrowth of harmful bacteria, potential pathogens, or frank pathogens, sometimes an overgrowth of yeast or even of parasites. What's important is that the balance of the community impacts the regulation of the tight junctions, and then that's what can lead to our increased intestinal permeability. So you can interestingly have dysbiosis in your gut without having any GI symptoms at all, and that's actually pretty common. Some of the things that can contribute include stress. We'll talk about that some more. PPIs, because altering the acidity of the chyme as it's leaving the stomach alters the control mechanism for the microbes in the small intestine. Gut infections that I've mentioned, diet, and then chronic constipation is another symptom or cause of dysbiosis. So both sides of the equation for that one. So if we think about how we were designed, so we were designed with this in mind, that this eubiosis of the gut microbiome would have this symbiotic relationship with our physiological and our immune tone. But what happens in our modern world is with our genetic predispositions, what we come equipped to do well, what we come less equipped to do well, Our infections over time, our diet, our lifestyle habits, our exposome, all of these things can create a reversible or temporary imbalance in the gut microbiome. And that creates a crosstalk for this transient and low-grade inflammation. So intermittent periods of low-grade inflammation. As this continues on, we can develop then into this sustained alteration of the gut microbiome, like what Dr. Pero was talking about in the terms of the different conditions and what the microbial composition of the microbiome looks like in those different conditions. That's our dysbiotic state. And that can create this low-grade sustained inflammation. And that sustained inflammation is a stressor on the microbiome. And then the imbalance in the microbiome leads to more sustained low-grade inflammation. So we end up in this cycle of chronic inflammation. What does it look like in an impoverished ecosystem? So this is an intestinal dysbiosis where we have a loss of diversity, so whether that's repeated antibiotic courses, for example, or a standard American diet that's low in prebiotic fiber, that is going to lead to the situation where we have our loss of microbial diversity. And with the loss of diversity, we see this increase in gut-related autoimmune diseases, Crohn's disease, ulcerative colitis, type 1 diabetes, celiac disease, even MS, and Parkinson's disease, which is also a gut-related degenerative condition. When we look at, for example, subjects with severe obesity, about 75% of those with severe obesity have low microbial gene diversity. So we have this interplay between the management and the maintenance of the body composition being related to the maintenance of the microbiome composition. My timer isn't going here if you want me. Okay, all right. So hang on. Okay. So our toxic exposure, I do a lot of work in environmental medicine, and so it's all about the exposome, and when we look at all the variety of exposures that we have on a daily basis, urban air pollution with carbon monoxide, sulfur dioxide, nitrogen dioxide, ozone, benzene, all of these different chemicals, plus significant amounts of heavy metals. I actually live in Northern California, right where gold was discovered, and so there was a lot of mining in our area. There's a lot of mercury, and when we have the forest fires sweep through, we have a dramatic increase in the aerosolized mercury in the air. So all of these different things, not just the smoke contaminants, but also the heavy metals that can be released from the trees because the trees take it up from the water. We also see that that air pollution has been shown to kill 10 times more people than motor vehicle accidents. In one study, about a third of strokes have been linked to air pollution, which is a really interesting thing to ponder. There are about 82,000 synthetic chemicals that with the Toxic Substances Act in 1976, I believe, they grandfathered in over 80,000 chemicals with no safety testing. So they were presumed to be safe until they were proven otherwise. So that's a lot of chemicals that weren't around 200 years ago. And we also know that of those 82,000, about 4,000 of those are produced in what's called high production volume. That means more than 1 million pounds of that particular chemical are either produced in the United States or imported into the United States. 4,000 over 1 million pounds. We also know from the study that the Environmental Working Group commissioned a few years back that newborns were found to have approximately on average 287 chemicals in the cord blood. So that's 287 chemicals circulating in them before they were even born. So we start our exposome even back then, even while we're a fetus. And then right now, only less than 1% of farmland is organic, less than 1%. And that, I think what makes that really remarkable to me is as early as the 1900s, 100% of farmland was organic because we didn't have any industrialized fertilizers, we didn't have industrialized pesticides or herbicides. And so in under 200 years, we've completely reversed our exposures to all of these chemicals. So how exactly does the diet impact the microbiome? We know that the Western diet, sometimes called the standard American diet or the SAD diet, is very low in fiber, it's low in PUFAs like the omega-3s, and we also see a high calorie density with a low nutrient content, and we have high in saturated fats and trans fats and lots of food additives that we'll look at here in a moment. But we have kind of these two main pathways. We have the presumably microbiota independent things like increasing adiposity, high cholesterol, insulin resistance. And then we have our microbiota dependent effects systemically with altering the microbiome, changing those metabolic byproducts that Dr. Pero talked about, and increasing our lipopolysaccharides, our trimethylamine production through the microbiome. These also independently affect these same outcomes. And then on the mucosal effect side, low fiber, so low prebiotic fiber, low food for the microbiome, and a high number of emulsifiers leads to a thinning of this mucus layer, which alters the biofilm, the healthy biofilm environment that should be along that intestinal lining. If we're looking here specifically at the colonocytes, these short-chain fatty acids are the obligate fuel for the colonocytes. So if you have a decreased production of short-chain fatty acids, you have a decrease in fuel source, in energy for the colonocytes. That means that you're going to have less epithelial barrier function, you're going to have less protection, and you're going to see a decrease in immune tolerance. So thinking back to the slide I showed a couple of clicks ago on the autoimmune diseases, gut-related autoimmune diseases, what type of cell do you think is present in high amounts in the colonic lamina propria? Which ones are related to autoimmunity? The Th17 lymphocytes. So those Th17 cells are present in high amounts in the colon epithelial lining, and if we have loss of barrier integrity in the colon, because we spend a lot of time talking about it in the small intestine, but in the colon, then that's going to increase the exposure of all of those peptides to those Th17 cells and increase our risk of some of these autoimmune diseases. So this is a busy slide, especially for those of you on this half of the room, but let me just walk you through it here. So our ultra-processed foods aren't just calorie-dense and nutrient-poor. They're also full of lots of different food additives. So just to orient you, this section is artificial sweeteners, this section is artificial emulsifiers, food colorants, food preservatives, and other items. And every single one of them, except for one, the emulsifier, the glycerol emulsifier, every single one of them decreases bacterial diversity. Every one. And if you look at the artificial sweeteners, you can see that we have increased permeability across the board. In the artificial emulsifiers, we see a decrease in the short-chain fatty acids, in mucus production, and in something that I can't read without my glasses, mucin 2 expression. And then over here in the food colorants, we also see decreased mucus production and gut barrier impairment. So all of these additives are adding to the burden, increasing our risk of intestinal permeability. When we look at the things that are the strongest proponents of the production of zonulin, so zonulin is the cell signal molecule that opens the gates for the tight junctions in the intestinal lining. What are the two strongest triggers of zonulin release? Any takers? That's a good guess. Gluten. Yes, in every person, not just in gluten-sensitive people, gluten is one of the most powerful proponents of zonulin production, and the other is LPS, endotoxin, lipopolysaccharide. So again, when you think about what are the setups that are going to lead us towards increased intestinal permeability, we have our food choices, and then we have our bacterial composition. And the Bacteroides species, which tend to predominate in dysbiosis, are high producers of endotoxin. So here we're looking back again at the overuse of antibiotics. And of course, when we think about antibiotic overuse, the first thing we think of is antibiotic resistance. But there's so much more to the antibiotic story. We also see changes because of the alteration in diversity. And when we talk about the alteration in diversity, I want you to be thinking about that every specific type of microbe has a job. So it's like a big community. So they all have different jobs. They don't do the same thing. And so when we're losing diversity, it's like we're losing the trash man, and we're losing I'm from California, so the PG&E, the electrical company repair guy, right? We're losing members of the community. When we see a decrease in fermentation because of the change in the microbes, we see a decrease in short-chain fatty acids. We also see changes in the metabolites and the enzyme activity that the gut microbes are supposed to produce. And we see an increase in some of the... Am I the only one that's having a hard time seeing these slides? Okay. If anyone can read, my biotransformation. There we go. It's super tiny for me here too. So the biotransformation of the liver, we have this kind of cross-communication between the microbes, what's being absorbed through the intestinal epithelium, what's being filtered by the liver, and then the biotransformation that has to occur for any of those chemical toxins, for example, or mycotoxins, for example. So we have this continued circulation, or enterohepatic circulation. And then over here on this side, the modifications in the biofilm. So if you've heard me speak before, you've likely heard me talk about the endothelial glycocalyx. I speak a lot about cardiovascular health. And we have this lining of the blood vessels that acts as part of the protective barrier for the selective permeability. And we have the same thing in the gut. So this biofilm, which can become dysfunctional, initially is an enteric glycocalyx. So that's the next layer on top of the microvilli, which creates a layer of protection. And in order for that to be healthy, the microbiome has to be healthy. So you just start to see this cycle of how things interplay. So dysbiosis does not just impact the gut. It doesn't just impact intestinal barrier integrity. It affects every single organ system. Because of my work in dementia reversal, I spend a lot of time on the gut-brain axis, and we see a significant amount of interplay through that enteric nervous system, through the vagal nerve afferents, of crosstalk between the cytokines and the cell signal messengers that are being produced by the gut microbiome, and then how that is changing what's happening in the brain. And we were talking about microbiomes at all of our epithelial surfaces. We talked about the oronasal and the gut and the vagina, but the brain actually has a microbiome. So there was just a study published this fall, and it looked at control brains versus brains of people who had died from Alzheimer's, from Parkinson's disease, and one other neurodegenerative condition. And what was super fascinating is that the control microbiome of the brain was predominantly oral pathogens, or oral microbes, not necessarily pathogens, oral microbes. So it was not the same as the microbial composition of the blood, which was different. So it's not just a diffusion of the oral microbiome into the brain. There is a gated mechanism, a controlled mechanism for the composition of the microbiome in the brain. And what we found in the hippocampus and the thalamus of the Alzheimer's sufferers is that they had a significant increase in what's called Cutibacterium species, which is a commensal species that can colonize the nose. So really fascinating stuff, like everything we thought that was sterile. I went to med school a lot longer ago than you did, 30 years ago. Everything that we thought was sterile, the placenta, the bladder, the brain, the blood, it's not. None of it is sterile. It's just not infected. It's not an overwhelming amount of bacteria. But we have these interfaces with the liver, with the lung, with the heart and the entire vascular system. And all of these have an impact on the health of the entire system. So the gut-brain axis has a significant contribution to mood disorders. We have a huge amount of our production of 5-HTP, about 90% is in the gut enterochromaffin cells and in the microbiome itself. That leads to a preponderance of our serotonin also being produced in the gut. And when we have dysbiosis and increased permeability, we see an increase of lipopolysaccharide, LPS, entering the brain, breaching the blood-brain barrier, stimulating the microglial and the astroglial cells in the brain to create that inflammatory cascade that leads to neurodegenerative disease. We also see, like we've talked about through the vagal nerve, this crosstalk between the intestinal epithelium and the enteric nervous system and the brain. We see the stimulation through the hypothalamus of ACTH and CRH leading to cortisol. We know that stress activates this same pathway, and cortisol changes your microbiome. It changes the composition, and that leads to our dysbiosis and permeability. So we have all of these cycles happening within the system. So we talked about the tight junctions. We talked about zonulin as the most important regulator, the gatekeeper of the tight junctions, and that the microbiome is important in regulating zonulin and tight junctions. And so are what two things? LPS, gluten. All right, good job, team. So intestinal permeability, where the gut lumen, or the antigens within the gut lumen, get access beneath the epithelial, that one-celled layer of the epithelium, into the subepithelium where we have the dendritic cells and we have the other lymphocytes in that lamina propria layer. That triggers our immune and inflammatory response. It increases oxidative stress. It increases chronic inflammation. This is super interesting. I literally just learned this this past week at the American Academy of Environmental Medicine. But LPS, when it's produced in the gut lining, when it's produced in the gut lumen, and then it gets across the gut lining, it deposits in the lining. And you can stain for that pathologically and see LPS. So it stays in the lining. It's not necessarily transient. So the more LPS that's produced, the more intestinal permeability that we have, the more likely we are to have a significant burden of LPS that's in the lining itself, perpetuating that chronic inflammation. I thought that was really fascinating. So stress, I mentioned on the previous slide with the diagram that this alters our gut flora. Sometimes we see digestive symptoms with stress like IBS or loose stool, diarrhea, for example. We also know that there's a direct action on the permeability. I talked about altering the stomach acid and how that impacts what bacteria survive the small intestine or survive our consumption through the stomach. When we are under a lot of stress, we typically think of stress as increasing stomach acid, right? Like the ulcer, the heartburn, that type of thing. But in reality, what we most often see is a decrease in acidity, an elevation of the pH. And what happens when you have that higher pH is that it doesn't send the signal to the lower esophageal sphincter to close it down. So you get reflux of stomach contents that are maybe pH 3 or 4 instead of 1 or 2, which is relatively more basic, but it's still acidic enough to give us the sensation, the experience of heartburn. But the problem is really the diminishment of acid production by that stress response. Cortisol also drops your secretory IgA, so at all of your intestinal barriers, that IgA is the first line of defense. And then, of course, for all of those reasons, it increases our risk of dysbiosis, and then that whole cycle that we've been talking about. Stress also activates an innate immune response, so just on its own, increasing the potential for inflammation. And what was really interesting is that perceived stress scales are associated with reductions in executive function, in working memory, in non-demented older adults. So just stress affects cognitive performance. And then there's also the mechanism for neuroinflammation via the toll-like receptors, and then that has to do with what we were talking about with the circulating toxins, for example, at the blood-brain barrier, breaching the blood-brain barrier, stimulating the astroglia and microglia via those toll-like receptors. So this is just more of a schematic of the stress inflammatory response. So we know all of these things with HPA hyperactivity, with that stress. We're used to thinking about these things: irritability, racing thoughts, poor sleep, altered appetite, changes in libido, somatic symptoms. And then depending on kind of the wiring of the person, that can lead more towards anxiety, it can lead more towards depression, and then always leading to these physiologic responses, which are part of what stimulate all layers of our inflammation and our chronic inflammatory response. So interestingly, our neurotransmitters, as I mentioned earlier, are very predominantly produced in the gut. And these are just some of the species that are associated with production of our various neurotransmitters. And it's really interesting that for GABA, for example, gamma-aminobutyric acid, that's our calming neurotransmitter, anti-inflammatory in the brain. And that is produced in large part by these lactobacillus and bifidobacterium species and Akkermansia. So really interesting combination of microbes that are responsible for that calming response, all of which are also associated with healthier estrogen metabolism. So you guys have these slides. The next couple of slides are a little information dense, and I definitely want to have time to go through our cases. In that impoverished ecosystem, so deficiency dysbiosis or lack of diversity, I'm going to start us in the middle here. So we know that these imbalances are clearly linked to mental health conditions. LPS-induced inflammation disrupts our neuronal integrity and specifically seems to impair the dopaminergic neurons, so our reward centers. Anxiety is associated also with decreased diversity and reduced production of short-chain fatty acids. When we think about these items that are termed psychobiotics, these are probiotics, beneficial bacteria, that influence our mental health by modulating the other microbes in the gut environment. And there are even these extracellular vesicles from these psychobiotic bacteria that can interact with brain processes. Dysbiosis also impacts tryptophan metabolism, favoring the pro-inflammatory kynurenine pathway over the serotonin pathway. It also increases cytokines and further affects neurotransmitters and neuroinflammation. So that really important interplay between the composition of the microbiome and mental health. Our sex hormones, Dr. Pero did a great job of really going through these for us already, so I'll just point out a couple of interesting things. Well, first of all, if you look at an image of the density of estrogen receptors in the brain, the concentration of receptors in the hypothalamus, in the hippocampus, in the thalamus, for example, all correlate very highly with the estrogen receptor network. So those are the same places that we see atrophy in the Alzheimer's brain. They're the same locations where we have a high density of estrogen receptors. Estradiol also promotes GLP-1 production in those endocrine cells. That influences our sense of well-being, satiety, stress response. Progesterone, interestingly, protects our enteric nervous system neurons. It also promotes neuroplasticity and resilience under stress. Progesterone is another calming hormone. Testosterone modulates neuroinflammation. Androgens inhibit mast cell activity. Increased mast cell activity is associated with a lot of neurodegenerative disease, so that interplay is important. And the difference in the estrogen receptor expression and activity also can be seen to underlie some sex-specific differences in IBS, for example, in visceral pain, and also in stress response. So I always choke a little bit on the word aging, partly because, we're all doing it, but also because what is aging really? I mean, it's really just an accumulation of your exposome. It's not, we do have clock genes. We are programmed to have a specific timeline to our lives, but much of what we see and experience as aging is really inflammaging, that term that's been coined, and that inflammaging comes from all of the things that we've talked about in the exposome. So when we think about how the gut microbiome regulates immune and cognitive health, there was a really interesting study that was done using a fecal microbial transplant. They took young mice, they took the poop from young mice, and they transplanted it into the old mice. And what they saw was that the old mice got rejuvenated simply from the fecal microbiota transplant. And the same was true in reverse. When they took the poop from the old mice and put it into the young mice aged more quickly and exhibited characteristics of cognitive decline. So really fascinating. The microglia are arguably one of the most important pathways of neurodegeneration. And we see that these types of protein aggregates, like beta amyloid, for example, or within the neuron, tau tangling, can disrupt the autophagy of the microglia and also of the neuron itself. Disease-associated microglia are associated with increased TREM2 activity. TREM2 is a genetically determined protein expression, and so if you come equipped with a TREM2 mutation, you have a higher amount of inflammation in your brain, or you're predisposed to a higher amount of inflammation in your brain. And then the activated microglia also spread, so there's a spread of amyloid and of tau that result from the activated microglia. So it's again like the other cycle that we saw in the gut microbiome, that once we have chronic low-grade inflammation, then that stimulates the cycle of continued microglial activation, astroglial activation, and continued neurodegenerative disease. And here we are. Women are more vulnerable to the effects of all of these things. Women have a 33% higher likelihood of anxiety. They have a 21% increased incidence of depression. And 2 out of every 3 Alzheimer's brains belong to women. So 2/3 of Alzheimer's patients are women. And 75% of caregivers are women. So we have 6 million people in this country that are affected by Alzheimer's or dementia, the broader term, and the majority of those are all women. We went through how the hormonal imbalances can lead to some of our anxiety, not just through our dysregulated cortisol, through insulin resistance increasing the chronic inflammation, that perpetuating our chronic inflammatory response. Thyroid dysfunction, both low or high, can lead to anxiety, low estrogen, low progesterone, and the sex hormones and neurotransmitters in the brain also need to interact, and we talked about the density of the estrogen receptor network. So many mechanisms for anxiety through this gut-brain hormone axis. Estrogen signaling is really interesting because we have the two main estrogen receptor types. We have the estrogen receptor alpha and beta. They lead to cell proliferation and differentiation, obviously mammary gland development, and then potentially to increased cancer risk, although that's questionable if that's a direct line. We see that phytoestrogens can also lead to similar tissue responses. Many of the phytoestrogens preferentially attach to the ER beta receptor. So we get more of the downregulation of the stimulating alpha activation. The ER receptor beta is also anti-proliferative, anti-inflammatory, and increases BDNF, which is very important. ER alpha is excitatory. It is a little bit more pro-inflammatory, but when you're looking at that estrogen receptor density in the brain, you need to have both. It's always about balance. So the way we were designed was for inflammation to be protective. Obviously, we weren't designed for inflammation to be our undoing like it is in our modern day. And at the time, it was avoidance of harm, it was sound the alarm, it was wound healing and fighting infection from running from the saber-toothed tiger or encountering specific pathogens, and all of that gave us a robust benefit for that inflammatory response. In fact, in African populations, the ApoE4 genotype is protective against certain types of infections. And so it was a beneficial change, it was a beneficial SNP. But then enter our modern world with more pathogens that arguably we've created by antibiotic resistance, for example. We have chronic stress, we have our load of environmental toxins that we talked about, and then we have this obesogenic environment in our ultra-processed foods. And then that leads us over here to higher infectious mortality, chronic inflammation, chronic conditions, increases in autoimmunity via all the mechanisms that we've reviewed. So inflammation is obviously important. It saves us, but chronic inflammation is super detrimental. And here's just our schematic of the inflammatory process. So we used to think that we had the triggering event, inflammation started, and then it just dissipated. It just went away on its own. You sprain your ankle, it gets swollen. As it heals, that inflammatory response, the swelling, the redness, the pain, all of that just dissipates. But what we now know, and that many of us are very well aware of because we're using these SPMs, is that turning off of inflammation, the inflammation resolution, is an active process mediated through those SPMs, the pro-resolving mediators, resolvins, that are helping us to decrease the inflammation that's present. And when we get stuck in that chronic inflammatory loop, it basically gets kind of worn out, if you will. So we just aren't actively producing those, and bringing in those specialized pro-resolving mediators can help us reinitiate that inflammation dissipation event. So here's the overview. Our genes determine our vulnerabilities, what we are well equipped to do, and what we are maybe challenged at being able to do. They then get exposed to all of our environmental toxins, medications, alcohol, infections, chronic stress, all of these things. That changes our epigenetic expression. And then that interacts also with our microbiome health, the diversity of our microbiome, determined by all of those experiences that we've talked about, the degree of dysbiosis that we end up with and that we fail to improve, that we fail to rectify. And then that leads to blood-brain barrier permeability and neuroinflammation. Leaky gut equals leaky brain. You cannot have a healthy brain without a healthy gut. They go hand in hand. It also leads to all variety of symptoms that we've talked about, and alters the way our body stores fat, the way we burn fat, and how we experience satiety when we're eating. So I have a couple of case studies that I wanted to go through. The first one, TK. So he's a 72-year-old male contractor. He's about 5'11", weighs 244, or weighed 244. His BMI is hovering right at obesity at 29.6, and as a result, he has all of the medical conditions that we would expect him to have at that body habitus: hypertension, prediabetes, GERD. He also has some ADD, so some potential wiring issues in his brain, but also potentially some neuroinflammatory issues. And then he has MCI, mild cognitive impairment. His medications, a couple of blood pressure medications, trazodone because he doesn't sleep well, hydrocodone at like 80 milligrams a day, and then sildenafil for his erectile dysfunction. He's on a handful of supplements that he started on his own. And then interestingly, his history includes this very high level of chemical exposure in his many years in construction and demolition. He worked a lot with wood glues and lacquers, and then he used to grind melamine for counters that has formaldehyde in it with no personal protective equipment for years. And then as a young child, he had at least 50 ear and throat infections, so treated with a massive amount of antibiotics. He was coming to me for cognitive decline over the last several years, and what was really bothersome to him was that he was forgetting the names of people that he knew, like his neighbors' kids that had grown up around him over the last 15, 18 years. He was losing his keys, his wallet, his phone all the time, every day. He said he had to look for something that he's misplaced multiple times a day. And then his wife said something interesting, that he didn't give normal eye contact anymore, that when you were talking to him, if he went to respond, he would look off at a blank wall or look at the ceiling because his brain couldn't process his visual experience of you and what he was trying to think about to say, which was really fascinating. And then he also had a positive head turning sign. Does anybody know what that is? That's when, I'm sorry? A positive head turning sign. So that's when you ask the patient the question and he turns to the wife to get the answer. Or every time he says something, he turns and says, "Is that right?" And it goes both ways. It's not a gender-specific thing. But it's a really interesting thing. If you pay attention to it, you'll start to notice it in your patients that have some cognitive symptoms. So here were his key findings. He had a MoCA, Montreal Cognitive Assessment, similar to the mini mental status exam, but more sensitive for mild cognitive impairment. That was teetering on Alzheimer's. About 18 is where we would call it Alzheimer's. His p-tau217, which is one of the new biomarkers for Alzheimer's disease, blood biomarker, was in that intermediate risk zone, so not quite diagnostic of Alzheimer's, but definitely showing an increase of amyloid beta pathology in the brain. Surprisingly, his hsCRP, while a little elevated, was not anywhere near what I was expecting it would be. His omegas were deficient. He had a fasting glucose of 95, fasting insulin that was quite high. He skimmed in below the pre-diabetic zone for his hemoglobin A1C, but we all know that that's a continuum. Between 5.2 and 5.7, there's a doubling of your cardiovascular risk. So don't think for a moment that being 5.6 keeps you in the safety zone just because you don't get the label. His lipid profile is a little bit off, but again, surprisingly, other than the expected hypertriglyceridemia, not anywhere near as bad as what I expected to see on that either, but his hormones are a disaster. He has essentially no detectable pregnenolone. That should be between about 100 and 250. His is 10. Pregnenolone itself is independently correlated with memory decline. So that's an important thing when we're working with patients with memory issues. His testosterone was extremely low, DHEA, sulfate. I was lazy, I didn't put the S on there, but it's always the sulfated version. And his free T was also low. And then he had very elevated levels of mycotoxins and, not unexpectedly, extremely high levels of chemical toxins. This is the summary of his GI assessment. So overall, doing pretty good, but bam, right there in the middle, dysbiosis is off the charts. So we did lifestyle modification, we started our precision medicine protocol with gut restoration, replenishing his nutrients, addressing all of the root causes of cognitive decline, adding the testosterone back in, and then some key supplements to address each of these areas that we're trying to work on over here. So an antioxidant-rich multi, like Phytomulti, a high-dose probiotic, like Ultraflora Biome Pro, detox support, omega-3 support, vitamin D, magnesium threonate, because the threonate form crosses the blood-brain barrier, and then a charcoal and clay binder to address his mycotoxin and toxin burden. So just 4 months into treatment, from arguably marginal Alzheimer's, he says, I'm remembering things 100% better. His wife is like, no. But he feels like he's remembering things 100% better. He has more energy, more stamina. He feels younger. He's still doing construction and working 12-hour days at 72. And his MoCA is now 26, which is right on the border of normal. So 12 months after enrollment, his symptoms of cognitive decline have resolved. I feel like my old self. I have boundless energy. His MoCA is normal, so we pulled him back from the brink of Alzheimer's and just needed to continue working on and supporting all of the things that we did. I see that I'm over time. Do you want me to do my second case real quick? Okay. Okay, so my second case, MM, a little bit on the opposite side of the spectrum, so I thought this would be an interesting contrast. So she's 66, yoga instructor, 5'2", 115 pounds, BMI of 20, so opposite. Her past medical history is pretty unremarkable other than osteoporosis and endometriosis. So just right now, knowing only that about her, what do you already know? Because she's had estrogen metabolism issues, and osteoporosis is a chronic degenerative disease of the bone. It develops because chronic inflammation stimulates the osteoclasts, right, the specialized bone breakdown macrophages to break down the bone, and chronic inflammation inhibits the activity of your osteoblasts. So it's an imbalance of the bone building cells and the bone breakdown cells, as we all know. But what we don't talk about in conventional medicine is the why. The why is the chronic inflammation, presumably through significant dysbiosis because she's had problems with her estrobolone as well. So she's on Fosamax. She's on, again, a handful of little supplements that she's done on her own. And she does have a dental implant after a failed root canal about 15 years ago, and she has 6 mercury amalgam fillings. So we already know about toxic burden, we know that she's got some infectious burden, and we also know that the oral microbiome is overrepresented in the brain microbiome. So she'd had cognitive decline for about 2 years, mostly difficulty and anxiety with learning new information, following instructions. During the pandemic, she actually had to learn a new job and was super anxious that she wasn't going to be able to remember how she was supposed to do things, to be able to follow the instructions that she'd been given. So some of that emblematic of executive function decline. She was having a hard time remembering faces. And at yoga, as a yoga instructor for a couple of decades, she was having a hard time remembering the new names. And that was really anxiety-provoking for her because she'd always been able to see that someone was new in her class, to meet them, and then to be able to greet them the next time they came. So that was bothering her. She also could no longer consistently remember her right from her left when she was teaching. So she would have to say, okay, now on the other side, and she'd say, okay, start on the right, and then she would watch to see what people did. So that was crazy change for her. And then she also couldn't remember if she'd already done a sequence on the other side when she was teaching. So lots of things that were affecting her quality of life. Her key findings, MoCA of 21, so squarely in the mild cognitive impairment zone. She also had a borderline elevated ANA, 1 to 80. So what do you already know? Specifically, leaky gut. Specifically leaky gut, because leaky gut is what leads us to autoimmunity, because where those peptides get across and they stimulate the immune cells, and then we develop the autoantibodies and we have the failure of the checkpoints. She also had low omegas, and then interesting, these F2 isoprostanes, these are a measure of oxidized cell membrane components, and hers were really, really high. So less than 0.86 is normal. She was 3 times that and had a lot of oxidative stress. Her hormones were also not good. She had very low pregnenolone. She had unmeasurable estrogen. Of course, she's postmenopausal, so that's not surprising, but when you think about the estrogen receptor network that her brain is crying out for stimulation to, she had nothing to give. No detectable progesterone, testosterone, free testo low. Her vitamin D was deficient. She too had very elevated levels of mycotoxins, and specifically the trichothecenes, which are such potent neuroinflammatory agents, neurotoxic agents, that they are used in biological weapons as nerve agents. And then gliotoxin, which as its name says, specifically inflames the microglia and the astroglia. So she had very neurotoxic mycotoxins in her system. She also had very high levels of chemical toxins. And then she had 4 risk pathogens in her oral microbiome. This was her GI assessment. So she didn't have quite as severe a dysbiosis, but she had much more impact on her short-chain fatty acids, and she had some potential pathogens and also some yeast in her gut microbiome. So similar intervention, just lifestyle modification. We use a ketogenic food plan when we're treating cognitive decline, exercise, gut restoration, nutrient replenishment. Again, focusing on all of the root causes of cognitive decline. We added hormone support, started her on low-dose naltrexone to modulate her immune response. She needed some thyroid medication, and then Welchol can also be used as a binder, specifically in mycotoxin. It also binds to other chemical toxins too. And then we had our key supplements that were added. She also had some L-glutamine in her mix. And 4 months into treatment, she now is no longer having the right-left issues in yoga class. She hasn't had any at all. She wasn't feeling anxious about learning anymore. And she had actually even decided to pick up a class in travel Italian. But her MOCA was still 21. So it doesn't always reflect the changes that we can see in the quality of life improvements. But 12 months after her enrollment, she could remember very complicated yoga sequences without any notes now. Her MOCA is completely normal at 28. In fact, she bought herself a new Mercedes as a reward for all her hard work, and her personalized license plate is MCI2NL, MCI to normal. That's what we can accomplish when we address the gut, when we address the exposome, and we pay attention to all of those things. Thank you.